This is on biomarkers of aging. And before I introduce our panel, I want to mention something that I think most of the folks who are attending may know about, which is that about a month ago, we held a webinar on this topic with this precise panel, along with Luigi Ferrucci of the National Institutes on Aging and Bob Temple from the FDA. Now, we're not going to, hopefully, we're able to build on that learning. We're not going to recapitulate that webinar. So I'll be asking the panel to digest and take into account what we learned and to proceed beyond that. But I guess I can summarize a little bit that Bob Temple was very supportive of the idea of biomarkers for guiding hypothesis generation, for enriching study populations, for selecting candidates for further study, etc. But on the $64,000 question of what it would take to promote them to surrogate markers and registrable endpoints, he pointed out that the industry needs to demonstrate this and to validate this in intervention trials first, and sort of not the other way around, to use the biomarkers to accelerate and hack or shortcut intervention trials. One of the examples that came up was the TAME trial, and Bob was very happy to say, hey, come back with a successful trial in 5 years and let's talk, which is good, but we want to know whether there's something more that we can do about that. Recall that biomarkers are laboratory metrics, biochemical results, and they are asserted to be sufficiently established to predict meaningful clinical benefit that moves the needle. And they could provide for the basis for a conditional or an unconditional approval. Conditional means that you'd have to have some phase 4 studies to validate after approval. And historically, surrogate markers like blood pressure, cholesterol, hemoglobin, A1C, viral load, intraocular pressure, as predictors of stroke or heart failure, diabetes, HIV, glaucoma, took decades of emerging scientific and medical consensus before they were accepted as regulatory endpoints. So on the one hand, the September webinar served as a useful reminder to the geroscience community that just because biomarkers are getting better doesn't mean that the FDA is going to strike their foreheads with their open palm and say, yeah, let's use this for regulatory endpoints. But also, we recognize that this is history's any guide. It's the start of a decade or several decades long conversation before they could be adopted. But we also understood that we as an industry would have to buckle down and perform and demonstrate the intervention trials. And also recognize that there were certain areas, other areas, mechanism, causation, modeling, predictability, and others that we could work on to support the substantial evidence that the agency will need to be able to adopt them as regulatory endpoints. Now, during day one, yesterday, there were a number of references to the openness of the FDA. Dr. Peter Stein said it, Joan Manick, Jay Olshansky reported it as part of their personal experience. The NIA has been conducting a number of conversations with the FDA about sharing geroscience approaches. So rather than repeating the conversations from September, hopefully we can build on this. And we're going to start with statements from our panelists, Steve, Morgan, and Polina, in that order. So Steve Horvath, please take it away.
Yeah, hello everybody. This is of course a session on biomarkers. So we will talk about the science, the latest biomarkers. I'm a professor at UCLA. My research area surrounds molecular biomarkers of aging, in particular epigenetic biomarkers of aging. Here's my opinion on biomarkers. We need solutions now. Actually, we needed them yesterday. We simply don't have 10 years of time to wait. So many of us are heavily involved in studying biomarkers. It's a burgeoning field. One could have organized a 2-day workshop just on biomarkers. So, and I'll stop with that.
Sorry, we have to unmute to speak, just a memo to self and to Morgan. If you are unmuted, could you maybe, if you have any opening remarks, please do before we launch into the intra-panel discussion.
Okay. Yeah, I think I agree with Steve. There's been tremendous work in the biomarker field, but I do think we're still not quite where we need to be yet. So I think there are a number of things that, we need to improve on. So number one, we need to show that we have good kind of reliability in these biomarkers we're measuring. So if I measured someone twice, would I get the same answer? This is going to be really important when we're doing these intervention trials or making any sort of assessment at the end of one level. The other thing I think that is really critical is also understanding the validity of these biomarkers. So what are we actually trying to capture? So there's been a lot of biomarkers trained to predict things like chronological age, but actually the more robust and more powerful algorithms you use to better predict chronological age, perhaps the worst biomarker you end up with. We do not want a perfect predictor of chronological age. The actual measure we're trying to capture is your divergence from your chronological age in terms of your biological profile. So I think there needs to be kind of a reframing in what the actual goals of biomarkers of aging need to be.
And Paulina?
Thank you. So I guess I will agree with both Professor Steve Horvath and Professor Morgan Levine on that. Indeed, the biomarkers of aging is already an emerged field with a lot of studies, a lot of publications, and a lot of discoveries made in recent years. Yet some of the discoveries are not ready to be translated into clinical practice. At the same time, I want to draw your attention that those aging clocks we are talking about, or those markers of aging we are talking about, they're mainly computational models. So it's not something we just measure in blood, but it's something we analyze computationally first before we get our predictions. And because they're particularly powerful, because of that, we can simulate the outcomes, and we can explore how our models that predict biological age will react to those simulations. And that's help us to generate hypotheses in aging space, and also explore how even humans will react to certain interventions without going to trials. So I think it's about time we actually it's about time for us to have a discussion with the FDA. I think because we have such an exceptional tools in our hands, probably what we need to do is actually have a DA to lead us into I don't know, maybe create some sort of initiative around biomarkers, and that will actually help us to drive them in a practice. So that's what I think is the most important. Probably not validation, but actually having FDA to lead this discussion.
Well, I appreciate the quick round, lightning round of comments, but I believe at least some of you had some slide decks. I think they came in rather recently, so I don't know whether they're ready at the central location, but in the order, Steve, Morgan, and Paulina, to the extent you have slide decks you want to talk to, would you please share your screen and make sure that you're unmuted and your camera's on. I see that Steve is putting on his slides, so please take it away.
Let's see. I hope you can see my slides. Yeah.
Yep.
I will talk about epigenetic clocks. Before I do that, I want to say the following. In the biomarker field, we would like to have biomarkers for all hallmarks of aging. Anything from mitochondrial function, autophagy, and so on and so on. There's one hallmark of aging where biomarkers have been developed with success, and that is epigenetics, especially DNA methylation. I give you a mini history. This field started in 1967 with a Russian researcher Berdyshev showing that global methylation decreased in the humpback and salmon. Then there were decades of beautiful work where, especially in the field of cancer, people characterized specific genomic regions that gain methylation with aging or lose methylation with aging. for example, islands and bivalent chromatin gains methylation with aging. For me, a personal milestone was the development of the Illumina array, in the sense of that's where my own research started with this platform. I was, of course, not involved in the development of the array, but this array facilitated large-scale epidemiological studies and cancer studies. Our first epigenetic clock was published in 2011. It was an estimator of chronologic age based on saliva. And
Steve,
at the
I'm sorry
time,
to interrupt,
yeah.
but part of your slides are cut off on the right. I don't know whether you can put it in full presentation or something like that. We can see most of your slide, but there's some text that's cut off on the right. Is there anything you can do to manipulate or reload it? Sorry for the interruption.
Yeah, let's see. I mean, I could do the following.
If you maybe you could minimize your left hand, the thumbprint, so that you could increase the size of your. There you go, or even more, because you know what your slides are going to be on the left column. Just maximize. That's.
Maybe I'll start. Is that good from you? Mm-hmm.
We can read it.
Okay.
That's a little too large. Somewhere in
Okay.
between. Okay.
How about that? Mm-hmm. Okay. Now, this brings me to the definition of the term epigenetic clock. In the past, people talked about epigenetic clocks as biological mechanisms, but we now have a precise technical definition. An epigenetic clock is a multivariate estimator of age or, importantly, mortality risk. So epigenetic clocks don't just estimate chronologic age. Some of them are constructed as mortality risk predictor. For example, Grimm age or pheno age. Back in 2012, an Italian team published a clock based on the single cytosine, and that single cytosine already achieved the correlation with age close to 0.9 or more. Of course, the so-called pan-tissue clock was applies to all tissues, which makes epigenetic clocks really stand out. You can have a single mathematical formula for estimating age based on all sources of DNA. And another remarkable property of these clocks is that they apply even to very young samples, for example, prenatal brain samples and so on. In 2015 and 16, several groups, independent groups, published large-scale meta-analyses that show that epigenetic clocks predict lifespan even after we adjust for a host of clinical and demographic covariates. 2017 saw the publication of several articles from different groups that described mouse epigenetic clocks that revealed that gold standard anti-aging interventions slow or reverse epigenetic aging. So in particular, caloric restriction is associated with slower epigenetic aging. Growth hormone receptor knockouts in mice are associated with slower epigenetic aging. So these are very robust findings. They have been validated by several groups. 2018 saw the publication of so-called second generation epigenetic clocks. They were trained to predict mortality and morbidity risk. So I mentioned pheno age, Morgan Levine published that, and also Grimm age from Acknowledgements. Grimm age has been carefully validated in many epidemiological studies. There are several citations that report cross-sectional associations with many measures of morbidity, anything from lung function, kidney function, even brain function, brain volumes. So also longitudinal studies show that the rate of change of grim age is associated with increased hazard of death. I'm sure you heard Greg Fahey talk about his exciting intervention to regenerate the thymus. And this study had a very nice side effect, which was epigenetic rejuvenation. So here I show you this the data from this phase 1 clinical trial, where in the course of the trial, after 12 months, we saw a substantial reduction of grim age, 2.5 years reduction after 12 months of this thymus regeneration treatment. So messages, epigenetic clocks are already being used in human clinical trials. I want to now shift gears and briefly talk about epigenetic clocks for mammals. So we leverage a new technology for mammalian methylation studies. This is a chip that looks at cytosines in highly conserved DNA sequences. This methylation rate applies to all mammalian species, mice, dogs, rats, and so on. It's well suited for studying aging and development, and it facilitated the construction of epigenetic clocks in over 50 species, all mammalian species. Example, here's an epigenetic clock for mice. It leads to very high accuracies in independent test data, many different exciting tissue sources, striatum, cortex, cerebellum, tails, ears, anything. Many aging researchers are interested in rapamycin. So in collaboration with Vadim Gladyshev and Rich Miller, we showed that rapamycin appears to have a weaker effect than caloric restriction on epigenetic aging rates. We have epigenetic clocks for dog breeds. And, but now I want to bring you to the third generation of epigenetic clocks. These are now clocks that apply to multiple species at the same time. So these, in other words, there's a single mathematical formula that allows you to estimate the age of multiple species. So a blood sample from human or a blood sample from a cat. So this is what we call a dual species epigenetic clock. So on the left-hand side, I show you human samples colored in orange and cat samples colored in green. And you may appreciate that we get very accurate clocks that, again, one formula that applies to cats and their owners. Similarly, we have such dual species clocks for the naked mole rat and humans. Again, very accurate. The right panel shows you the accuracy in the naked mole rat, correlation 0.95. But again, the same math formula applies also to humans. Similarly, we have such dual species clocks for rats and humans. And so why do we develop these clocks? It's all about increasing the chances that a treatment that reverses aging in a rat will translate to humans, right? Because if you use the same cytosines that respond to the treatments, cytosines that are in conserved regions of the genome, chances are that the same clock will also show a beneficial effect in humans. I briefly mention, we have built universal mammalian clocks that apply to over 160 species. I show you here on the upper panel the leave one out cross-validation estimate. And here the dots correspond to tissue samples from all these species that are listed on the right-hand side, anything from human to dolphins, rabbits, sheep, bats, and so on. So that's really, really, in my opinion, a miracle of methylation, that you can build biomarkers that apply to different tissues from different species. It's just mind-boggling, at least to me. Another topic is, does cytosine methylation relate to maximum lifespan across mammalian species? So we're not talking about chronologic age, we're talking about species differences. And the answer is yes. So we have actually epigenetic predictors of maximum lifespan. In other words, if you send us a blood sample and you don't tell us anything about the species, it's an unknown species that you found in the jungle, we can give you a rough estimate of the maximum lifespan of that species. Correlation is here 0.88. That brings us, of course, to a scientific question. Can we extend the maximum lifespan of our species by modulating DNA methylation levels? And so can we edit specific chromosomal regions where we modify methylation or other epigenetic marks to extend lifespan, for example? And also another question would be, what about slowing the rate of change of methylation across the lifespan? Maybe that helps us to extend the lifespan of our species. So what's the future? Our clocks require an improved mechanistic characterization. What are the causes of methylation changes? What are the consequences, the cellular consequences, the consequences on organ dysfunction? And also, we need to continue to optimize epigenetic biomarkers and complementary biomarkers for human clinical trials. We need to ensure that retest variability is low, that these clocks are strongly predictive of the outcome we care about. Is the outcome mortality? Is it multimorbidity? Are these specific diseases? So this will keep the field busy. In conclusion, human epigenetic clocks differ in predictive accuracy for mortality. The pan-tissue clock is not great. The Grim-Age clock is the best predictor of mortality in our hands. None of these biomarkers will replace standard clinical measures such as blood pressure, lipid levels, glucose measures, and so on. They will enhance them, but not replace them. And also, as you design your own study, you need to be careful what cell type you profile. Sometimes blood is the obvious choice, but suboptimal. Sometimes buccal epithelial cells are more informative, or you can think of other more inaccessible tissues. And I'll stop with the acknowledgement slide. Thank you.
Thank you, Steve. Throwing the mic over to Morgan, please. Please unmute yourself.
Okay, let me start sharing. Okay, so I'm Morgan Levine. I am an assistant professor at Yale University, and like Steve, I do a lot of work specifically looking at the epigenetic clocks, but also quantifying biological age in general. And I'm not going to spend a lot of time on this slide because I'm sure it's something everyone already knows, but there's an immediate need for actually being able to quantify the biological aging process, particularly in humans, because when it comes to doing intervention trials, using the traditional outcomes like morbidity, mortality are often not feasible due to time constraints. So this is really where we need to try to optimize what we call these biomarkers of aging, which are going to be useful proxies to actually assess the agedness of a sample. However, there's some issues with actually developing these. So number one is where do you even start? What kind of biomarker or what kind of variables do you actually use for this? So as we know, nearly everything actually changes with age, and it's almost an issue of having too many options to start with. And really what we need to start thinking about is which changes are actually the most informative for health. So yes, I could make a biomarker of age based on hair pigmentation, but is that actually going to tell me anything about future risk of morbidity and mortality, which is actually the ultimate goal of what we're trying to intervene in? So in my lab, we like to think of this in terms of biological levels of organization. So we actually think that aging is starting at these lower levels of biological organization due to a number of different reasons, whether it's kind of errors or entropy or damage. It could also be adaptations and compensations, which we need to distinguish from damage. But also there's also selection pressures at kind of the cellular versus the organismal level. And we think that over time, these changes at the lower levels of biological organization are going to propagate upwards. And this is going to, in terms of emergence, manifest as pathology or ultimately death and disease. So for this reason, we like to actually measure molecular aging. So Steve discussed epigenetic clocks a little bit, but just in general, what you can do is you can take a group of individuals, they might all be the same age, maybe 45 years old. You can take a sample, whether it's whole blood, saliva, tissue sample, and then we can actually measure DNA methylation levels as CpG sites across tens of thousands to often millions of these genomic locations, and then apply machine learning approaches to get out an estimated biological age. So perhaps 41, even though chronologically they may be 45. And what we expect is that your difference in your biological age and your chronological age should tell us something informative about your future health risks relative to other people your age. So in terms of biomarker goals, I mentioned this very briefly in kind of the opening remarks. I think there's a number of things we need to really focus on when trying to develop better biological age measures. So these come down to validity, reliability, modifiability, and mechanism focus. And I'll briefly just touch on each one of these. So first, validity. Again, we can develop biological age predictors using a number of different variables as inputs, but also predicting a number of different outcome variables. So most of these have actually prioritized chronological age predictors. Number one, because that's actually very widely available data. But actually, when we develop chronological age predictors, we're not actually prioritizing the most critical changes. You're just prioritizing what's going to give you the best chronological age prediction. And in many ways, when we use these very powerful, whether it's deep learning or machine learning approaches, we actually might be adjusting out the things that are leading to divergence between your predicted age and your actually observed age. You also have issues of mortality selection bias. So we actually should not get good chronological age prediction for individuals at the oldest old, because we actually think that they're a resilient subgroup of the original cohort. And actually, if anything, we should expect them to be underestimated. We should not expect good chronological age prediction. However, when it comes to doing these phenotype aging predictions, like Steve discussed with these second generation clocks, whether it be Grim age or the DNAM pheno age, we do think they capture more important information, but we are also at a disadvantage in that there's actually fewer training samples with this kind of information available, and it's not entirely clear which outcomes we should be training predictors of. The second important thing, especially if we're going to use these for evaluating interventions, is reliability. So we've actually recently found that a lot of the epigenetic clocks are actually not that reliable. So this is data from, I believe we had 8 individuals assessed 9 times using the exact same sample. So I take a blood sample or a saliva sample from someone, and I run it 9 different times. And the question is, do I get the same biological age prediction? And the answer is often no, which is really important if we're actually going to be assessing whether we get changes as a function of an intervention. So we've actually done, so this is just to acknowledge a conflict of interest. I'm a consultant for Elysium Health, and so this was in collaboration with them in developing their methylation measure called Index, where we actually did a lot of work in trying to correct out this technical noise. But I think this is something, as we move forward in developing these biomarkers, needs to be a main focus. So the next thing is actually developing mechanism-focused things. So the other issue in terms of the biological age measures and epigenetic clocks is they come across as very black box. We actually don't know what's contributing to the biological age estimates. So what my lab has actually started doing is trying to do more bottom-up approaches. So can we actually develop measures of specific cellular and molecular processes and then see whether these are actually driving some of the changes we see, whether it's in the overall epigenetic clocks or otherwise. So for this, I'll just briefly run through a very quick example where we actually made an epigenetic measure that tracks passaging in MEFs. So basically, we were able to make a really accurate prediction of population doubling. So this is what we're calling DNAM rep or replication. In these maps, we then looked at these in a variety of different tissues, so liver, lung, kidney, blood, adipose, and skeletal muscle, and found that in general, it also tracks age changes in all of these, although maybe not in skeletal muscle, which is not surprising given that it's postmitotic. We also showed, like a lot of the other epigenetic clocks, that this was responsive to things like caloric restriction. So we found a decrease in this epigenetic measure for calorically restricted animals. And we also found that it was reset like other clocks in terms of epigenetic reprogramming. And ultimately what we're finding is that actually this measure itself is driving a lot of what we see in terms of the general epigenetic clock. So now we're also working on measures for cellular senescence, mitochondrial dysfunction, and other hallmarks of aging. So finally, this is my last slide. I think another really important thing is understanding modifiability. So I'm not going to go too much into what this intervention was, but basically, like Steve showed with some of the interventions, can you apply these biomarkers of aging and actually track changes? But I think even beyond that, can we actually use the data from the interventions, whether it be things like the TAME trial or other geroprotective interventions, to actually develop better biomarkers? So can we actually find the cellular molecular changes that are mediating the association between whatever this intervention is and the decreases in morbidity or mortality risks? So can we actually find what's on that causal pathway? And with that, I just want to end by really emphasizing that I think aging clocks, regardless of how they're developed, can be really useful and powerful predictors for either clinical trials, basic biology, or risk stratification. However, we need to get beyond the black box and figure out which changes matter most for health. We also need to figure out how many clocks we need. So ideally, you won't want to just measure one biological age measure. And we also need to figure out which changes are actionable or modifiable. And with that, I just want to acknowledge my group, my collaborators, and my funding. Thank you.
Morgan, thank you so much. I'm throwing the mic over now to Paulina, who I think she's loading up her slides or sharing her screen. Thank you.
Right, so I guess you can see my screen now. My name is, I don't think I need to introduce myself one more time, but just to recap, I am a chief scientist at Epidemiology. We're a new company that focused on applying aging clocks and developing those. And a bit about the company, we were founded in Hong Kong 2019. Our main specialization is deep aging clocks. We use deep neural networks to design predictors of age. We try to publish as much as possible, also have several patents on those, and developing digital tools that integrate those aging clocks. A bit of a recap, both Morgan and Steve presented to you so-called epigenetic clocks. And of course, they're really powerful in terms of age prediction. At the same time, there are other data types that has been used to predict age, including transcriptomic data. And those advances in generation of multiple clocks was mainly led by accessibility of data in the public domain. So right now we have multiple amount of samples available for us to actually construct those aging clocks. At the same time, another revolution is happening. It's revolution in algorithms themselves. So the previous generation of aging clocks was mainly designed using linear models, or so-called shallow machine learning. But right now we have a completely new technology that is changing AI and actually generating incredible advances. It all started in 2014 with the ImageNet competition, where deep neural network were able to classify images way better than superhumans or superhuman in that case, who was actually trained to classify those. And since then, a lot of developments were made in terms of algorithms. So right now we have driverless cars, and we have also a revolution of deep learning in healthcare. Why it's happening right now? So most of those algorithms are not brand new. So most of those ideas, they came from 1980, but right now we actually have a way of training them. Before it was just a theory. Right now we computationally can do that. At the same time, we have a computing power to train them, and we have millions of samples because they're exceptionally data hungry. And what is happening in healthcare right now, since 2017, FDA approved 14 purely AI-based tools. So those were developed using different flavors of deep neural networks. And for most of them, FDA granted fast track approvals. And those are ranging from the arterial cardio MRI based heart interpretation, or to one of the recent approvals that was done for Apple for the atrial fibrillation detection. So most of the so people who own those Apple iWatch clocks, they now right now can actually use this technology. And of course, the deep learning entered the biomarker field as well. Since 2017, multiple aging clocks has been developed. They all, again, range in accuracy, but this we believe is mainly due to differences in data types. Just a bit about differences between different models. So shallow or linear models, they're just trying, as Steve presented, create those equations, just map the input parameters to the output by assigning a coefficient to each of their variable on input layer, while deep models are actually stacking multiple layers, and therefore they can do a lot of nonlinear transformations and capture really complex dependencies in the data. In data, for example, in between changes in blood markers or in methylation level and actual chronological age. And that's why they're particularly powerful, especially in analyzing biomedical data. But why do we need so many markers? So apparently they measure different things. That's a really nice study that was published in 2019 by Professor Dembelski, where he showed that if you analyze people's same chronological age, healthy people, you will see a significant variation in their biological age prediction. Also that different, even same data types, like different methylation clocks that were built using methylation data, show little or no correlation between each other. And at the same time, we see that clocks are ticking in the right direction. So people are getting older unless their own interventions, not only chronologically, but also biologically. So those clocks, let's say, are working in a direction we expect them to work. At the same time, they show no correlation between each other. So we would expect that those different clocks actually reflect completely different processes in the body. That's why in a company we are focused on a really diverse portfolio of aging clocks. So we have clocks that are built based on data from wearable devices, but also on omics data, including transcriptome and microbiome data. We started to work on clocks in 2013, and since then we published a lot of papers showing how deep learning can be applied in biological age prediction. And the first clock I just want to briefly show is the deep microbiome clock. So right now we can predict age of healthy individuals using their whole genome sequence data of their gut microbiome. We also were able to show that patients with type 1 diabetes actually have signs of acceleration. So they're predicted older compared to healthy peers same chronological age. But what's most important we can do with clocks is not only predicting biological age, but also explore how changes in input parameters affect biological age prediction. So this way we design those plots and we analyze the abundance of each of the species in gut with respect to their influence on age prediction, whether they are seropositive, those who accelerate the biological age prediction, or seronegative, those that decelerate. And what was interesting also to see when we analyzed it further, that not all SENA positive or SENA negative status, SENA negative species also have positive function in gut. So some of the SENA negative, the one that decelerate aging or decrease age prediction, are also associated with youth infections and so on. So that was an interesting observation. Another clock that we work on is the blood-based one. Here, not only we can predict age again, but also we can see that the predicted age is associated with mortality. So people who are predicted older, they have higher hazard ratio and lower life expectancy, and other way around. Smokers who are known to live shorter lifespans are also predicted older, actually twice as old compared to non-smoker peers, same chronological age. But we also can do with what we also can do with those clocks. We can actually explore how changes in the input parameters affect predictions, again, in the same manner that we do with microbial taxa. So just to summarize and recap, so of course, a lot of aging clocks has been proposed so far. And at the same time, we have in our hands completely new technology. FDA already recognized the potential of this technology because deep learning is just superior in terms of accuracy compared to other methods. So we believe that if we will have a clock that was built using this AI, so-called AI technology, it could be a completely different story to FDA. At the same time, we believe that those aging clocks are not only useful in prediction of biological age, but also in identifying variables that are associated with age the most. And that's why I want to draw your attention to this first point I made during the intro, that those models that we have, they're computational approaches, and that's why they can be used as tool to simulate the effects of even anti-aging interventions. So ideally, we could even run in silico clinical trials using those aging clocks without the need of going into really costly and time-consuming experiments. And then we can confirm those hypotheses that will generate, of course, in wet lab or in animals, but still, I guess it should accelerate and should be a completely different story to FDA in the end.
Terrific. Thank you so much, Polina. We have about a quarter hour left, and it's a little bit of a quandary how to divide up the time, but let me start first. The model for Metabesity is that this is a dinner salon. We're throwing together guests, experts in different domains, either subdomains or completely different functional domains. You've heard each other's presentations now. I think for an attendee such as myself, I would be interested in a sort of a curation. What burning questions do you have of each other based on what you heard from the others? Or if it gets to fisticuffs, sort of a tussle or a fight, Grimage versus Index versus something else, that makes for good television. Maybe you could spend a couple minutes to see how these different approaches in biomarkers are reading in each of your respective experts' minds. Whoever grabs the mic.
I'll start. The good thing is we are quantitative people, and everything will be decided by correlation coefficients or p-values. You know? So in this sense, this is not a political debate. What the field really needs are what I want to call data competitions. So data need to be generated, they need to be blinded, and then several groups make predictions on the data, then an independent jury unblinds it and just declares the winners. You know? And there's a wonderful tradition in the artificial intelligence community who conduct these kinds of competitions. I hope somebody will organize it for the biomarker field. If not, I will organize it, certainly for methylation. You know? So I've started a non-profit foundation called the Epigenetic Clock Development Foundation. The word development is important because we continue to develop new biomarkers, and I think we will organize that. Like Morgan mentioned, there's a need to evaluate biomarkers in terms of technical variability. We have generated, I want to say, 9 data sets with technical variants. GrimAge is very reliable, at least in these 9 data sets. But in general, that's what's needed. You know, independent juries need to evaluate these biomarkers. And basically, Morgan and I, we always have the same opinion. Whenever I hear her talk, I say yes, yes, yes. So I'm not sure whether Morgan agrees with me, but I certainly agree with
Let's
Morgan.
find out.
So it's a one-sided agreement, you know.
No, I mean, I agree with Steve. So I'll just say,
yeah. When it comes to the third talk, the deep learning methods, I will partially agree in the following sense. Deep learning methods are wonderful the minute you have very large data sets. And we can discuss what is a large data set. I want to say 100,000 observations. However, these tools can actually be inferior when you have a small data set. You know? So you could imagine you have 1,000 people for whom you have a very valuable data set. You want to build a biomarker. And in that case, nonlinear approaches can overfit. I'm not just talking idly. I want to say in my lab, we have carefully evaluated deep learning approaches in these smaller data sets. And there's a reason why we continue to use penalized regression model. They work better for these smaller data sets. And also, one shouldn't forget one important thing. Regression models give you a formula, so it enhances interpretability. You kind of know the covariates. By contrast, the so-called black box AI approaches can be a bit harder to interpret, you know. Now, that doesn't mean one shouldn't use AI black box approaches. I use them all the time in my research. They can have, as Paulina emphasized, they can lead to far more accurate estimates in the right data set. And I'll stop with that. Huh?
Any takers of the mic, Paulina or Morgan?
I mean, I'll reiterate, I agree completely with Steve. I'm very excited for the nonprofit to get up and running. I think what we really need is critical evaluation of all of the biomarkers that are being developed. There are so many that are being published. I mean, you see, you know, dozens of new ones every year. And I think the field really needs a way to critically evaluate them and prioritize them, because otherwise I think it's just going to saturate the field and actually, if anything, impair our ability to move forward and actually get these utilized in clinical trials. The one thing I want to also reiterate that Steve brought up a little bit, I'm not, again, ganging up on Paulina, but I do think, you know, the deep learning is really powerful when you have the right outcome that you're predicting. It's only as good as whatever you are trying to predict. And I mean, my concern in the field is about these chronological age predictions. I think there have been actually a few papers that have shown that you can apply these deep learning, I think from the group at Seoh, where you can apply the deep learning algorithms to get a chronological age prediction. You actually get a worse mortality predictor because you're getting such a accurate chronological age predictor that there's no divergence between chronological age and biological age at that point. But with that, I think, you know, I agree with a lot of what Steve was saying.
I'm going to give Paulina a chance to respond, but just a quick note to Steve. You said that this is quantitative and it's all impartial, it's subjective in some sense. But of course, you all made the point that different clocks and biomarkers measure different things and the field needs to decide for what purpose. And those are qualitative, not quantitative. But Paulina, please go ahead.
Right. So no, I totally agree with you, with Steve and Morgan, that we need to have certain, like maybe hackathon, or maybe, I don't know, some of the competition on aging clocks. But I also think what we need a set of metrics, standardized. It's not standardized yet, and probably some golden data sets for us to validate aging clocks. And one of the issues I see there is that we will definitely need mortality as one of those validations to see whether our predictions are associated with mortality and lifespan. And yet mortality is rather sensitive data. So I guess it will be a bit tricky to make it as public as possible. But at the same time, I guess there are ways how we can, limit access, satisfy the regulatory bodies, and still have it. At the same time, in terms of your comments on black boxes, I think I tend to disagree, because there are certain ways how you can extract weights from deep neural networks, again, by adding those penalized layers. And there are this type of networks that this type of model that called deep feature selection model will actually do that. So you have your input layer, and then you have a weighted layer, and then you have your hidden layers that are trained on to capture nonlinearities, but then your weighted layer is there, and you can still extract those coefficients and rank the input parameters by their importance. We do this all the time, and that's how we get those feature sets. There are also other ways how you can understand how the model is actually making predictions, and that's what we use to simulate the outcomes. I think that's really important, that's crucial, and that's, again, it's possible with deep neural networks purely. One of the issues with deep neural networks is that, as Horvath said, they tend to overfit, and the publication that Morgan referred to actually was experienced that. At the same time, you also have to have, it's not like it's easy to train them on simple, computers. You still have to have access to GPUs, and it will take a while to optimize them. So that's why we never do the development of a new clock with the deep neural networks first. We first go for something simple, just to understand that there are dependencies, at least there, or something is predictable. But apparently for some data types, not going deep learning is not even possible. For images, it's not going to work. So for any image-based clock, you just will have to go with the deep neural networks. That's a set true. For some of the data sets that you don't have a lot of samples, like, I don't know, 1,000 samples, well, that's a pity. Yeah, for sure, you will go for something linear. But then I think it's worth time in investing in developing of a deep neural network based model that can predict everything accurately. So yes, I guess all of us will have to unite and then come up with this this validation approach for the model that will be easy to use. It should be probably some library that we can install, all of us can install and can easily run on our newly developed models.
So this is a fascinating topic. We could spend all day talking about this, the technical aspects we just talked about, but something is sort of a follow-on from the September webinar. I think it was Carl Flager who mentioned this, or someone else who said there are like 5 clocks on the market today, and probably more and more. We've got members of presenting at the Emerging Company Showcase, and it's a couple of companies that are seeking to make a business out of clocks. How do we know about these, if you will, direct-to-consumer clocks? And Morgan, you participated in designing one for Elysium. Any comments about how the consumer is to judge and select amongst them? And it's complicated further by the fact of what are you doing it for? What you want to know is how much gas do you have in the tank, how many years left? I guess that's one question you could ask, but there are lots of other questions, as we've brought up, that you can ask. Comments?
The answer is easy for a scientist, So why? Because a scientist will look at peer-reviewed publications, right? Because many of the clocks have been used in studies, in dozens of papers, and you can just look up a few papers and say, oh wow, in this context it worked really well, in an independent data set, it predicted mortality and so on. So you look literally at peer-reviewed publications. Beyond it, it's hard to prioritize companies that offer different clocks. Personally, I'm really happy that there are many companies that offer clocks. Why? It gives the consumer a choice. Consumer can compare prices. I love that the prices are coming down. I caution people that you need to be careful of what is being measured, you know. This discussion, is it an estimate of chronologic age, or is it an indicator of biologic age, right? And as I mentioned, the clocks differ, and I'll stop with that.
Any others? Because we can move on to some questions, but.
I mean, I agree. I would really encourage the companies to publish, you know, maybe they don't have to put out, you know, the secret juice of what goes behind it, but give independent researchers a way to calculate whatever the clock is and evaluate it. So I think that's going to be very critical for actually figuring out which of these measures are actually valid and capturing what we want. But I think it is, I agree with Steve, it's great that there's multiple groups doing this now.
3 minutes left. Let's go to a question from Zidong 2. Have you looked at the maximum lifespan of human species predicted based on a methylation clock? It's got 3 upvotes, so I'll throw that out. Lightning round answers, please.
Yeah, we certainly have looked at it. It's our favorite mammal. Interestingly, these we feel these estimators of lifespan more often than not underestimate the ages of humans. You know, so I wish I could tell you that these clocks predict humans will live till 150. But when you think like a statistician, it becomes clear that it makes sense that these clocks underestimate the maximum lifespan. So think more, they estimate it to be maybe 110 or 100 even.
Carl Flager is asking, sort of at Morgan, will your index test be turned into blood, blood version? And he also makes an interesting observation, popularizing age clocks. Steve, to your point, you're happy that there are more of them. In general, is a great way to raise awareness of the field in the public. But Morgan, back to you for the specific question.
I think right now, I mean, I don't know. I'm just a consultant for Elysium, so I don't know exactly what their goal is for that. But I think right now we're doing the saliva test. Dried blood spots don't really work, so this would have to be a way for people to actually go in and get a venous blood draw. But it's definitely not out of the question. Although we are actually working on just making the saliva test much more accurate, like the blood test. It has to do with kind of a cell composition issue. So I think we can actually get the saliva test to mirror the blood test.
I apologize for the sorry, Steve, go ahead.
If you have blood samples, you definitely want to use the Grimm age clock. You know, I always tell people, I give them a money back guarantee that it will validate, you know. If you have an epidemiological study, you apply Grimm age, it will predict mortality. It's a very powerful predictor. But it only works in blood, not saliva.
Okay. One last shout out. You know, Steve and I were talking about activities of his foundation, and he's been thinking about looking at prior clinical trials that have looked at longevity-like endpoints, like the DPP, the diabetes protection, the DPP, and the SGLT2 inhibitor cardiovascular outcomes trials data sets. So if there are people who are out there listening who have ideas, want to get involved, think about funding for that kind of an effort to see if we can do some validation based on existing data sets rather than going on and creating new data sets, please contact us and we'll put everybody together. So at this, I want to thank the panel very much. We're actually doing another one of these at the Geroscience, the Gerontological Society of America pre-conference meeting on October 27th. It's a continuing conversation, and it's going to take 10 years. Steve, I'm sorry, it's going to take a long time to get this popularized and accepted by the regulatory authorities, et cetera. So thanks to the panel very much.